Cancer represents one of the most lethal diseases worldwide. Even in 21st century, cancer has remained one of the most challenging diseases to tackle with. Why cancer is so hard to treat? What happened when cancer becomes malignant? With the advancement in biomedical engineering, is there any tools that we can use nowadays to better study cancer accurately and efficiently? My name is Ting-Yuan Tu from Department of Biomedical Engineering at National Cheng University. Today, I'm very glad to share some of my lab works on studying the metastatic pattern for cancer drug development. So, as you might have known, cell, the most basic unit for all the living things, like your tissue, organ, human body, or all the animals, even plants, they are all made up of cells. So when the cells proliferates, usually it moves on, and also sometimes when it detects itself with certain damage or issue, it starts to sacrifice itself through committing suicide, a quote-unquote apoptosis. However, on the contrary, for cancer cell, it's not the way. Cancer cells can be caused by oncogenic substance, like chemicals or radiations. So when cancer start to have genetic mutations, they start to evolve. They have evolving with different genetics mutations. At a certain thresholds, they cannot stay on, and they become more malignant. They start to become more proliferative, become a cluster of cancer cells. So how to treat cancer? In the past centuries, there are three traditional ways, such as surgery, radiation therapy, and chemotherapy. In the recent decades, there are more new methods coming up, like stem cell therapy, hormone therapy, targeted therapy, and immunotherapy. So using targeted therapy as an example, a study done by Harvard, they studied the patient with BRAF mutation in melanoma. You can see the picture on the left. The patient was suffering very hard in melanoma before the treatment. However, when used targeted therapy, a RAF inhibitor, treating this patient, you can see after 15 weeks, this patient pretty much got recovered. Isn't that amazing? But the challenging part for the cancer is that the cancer can be resistant. The sad story for this patient is that after 16 weeks, the cancer starts to resist the drugs. The drugs means no longer useful. And after 23 weeks, the patient pretty much cannot hold on and passed away. So this becomes a major challenging for not only targeted therapy, but for a lot of therapy treating on cancer, that the cancer itself can resist the treatment. So if the treatment is not useful, can we do another treatment? Yes, we can do second-line treatment or third-line treatment. Even though we know that patient has certain mutation, however, it doesn't mean we have drugs to treat them. But why we don't have drugs to treat them? In order to understand this, we may need to see the drug development progress. This slide shows you different phases of drug development process. So you can see initially we will start with drug research and preclinical study, and then we move on to clinical trials. If we get lucky enough, after a few fails and success, we may move on to the final evaluation stage and get approved. How long does it take? It is not judged by months, weeks, easily it gets years. In order to get one drug approved, usually it takes more than 1.7 billion US dollars and over 10 years. But why developing a drug is so difficult? Maybe we can look at the first two phases. Because usually when the drug passes the first two phases with very successful outcome, but when it gets to the clinical trial, it failed miserably. So we need to look at how we do drug development for the first two phases. For the in vitro study, you can notice this is where the cells are grown. If you know this is the plastic we call Petri dish, we supplement the cells with nutrient and medium, so the cells are happy. When I first do cell culture, I always wonder what on earth the cells are growing on the plastics. Does there anywhere resemble our human body? This has been a major question in my thoughts. But it doesn't trouble me for too long. Then I realize, the reason is it works. It works is very important because you can see a lot of medical breakthroughs from Nobel laureates that used to use this platform to have many, many medical breakthroughs. And then if we move on to animal studies does recapitulate several parts of the human body. In fact, 92% of DNA are shared among human and mice. But new studies suggest that because the different use of the genes, that actually a lot of drugs work on the mice cannot be used for human. So this gives us a hint. What should we do? How can we move on? Is there a better model can be developed for drug research and the preclinical stage? In order to understand how to build a better model, I think we first have to look at the progression of carcinoma. So you can now see in the beginning, in the initial phase of the tumor progression, it is rather pure. Mostly are tumor cells alone, not many other cells involved. But as the tumor gets more malignant, you can see some cells involved, like fibroblasts or immune cells. Even in the later stage, you can start to see this tumor cells. The tumor itself cannot sustain the nutrient and oxygen in the core of the tumor, so they start to attract blood vessels into the tumor. This is the process we call angiogenesis. So how do we simulate this tumor microenvironment? Earlier this year, we used a microwave platform. We forced the cells to aggregate. Of course, these cells are cancer cells. So we force them to grow into tumor-like structures. Over a course of 4 days, we can see very improved cell-cell interaction and structural similarity of this in vitro tumor as compared to the avascular tumor in vivo. And because of this platform, it's made in the traditional cell culture plastic wares, so we can easily make hundreds to thousands of tumor in vitro. We also study these tumors on various cancers, such as bladder cancer, lung cancer, and liver cancer. We notice there does indeed significant difference in the permeability of the small molecule drugs in different types of cancers. We managed to use these in vitro tumors to get a very improved drug screening outcome as compared to the traditional culture method. We are very glad that we published this on Global Challenges and accepted as a cover story earlier this year. However, not only the tumor cell alone. As you can see, in the later stage, the cancer cells recruit more and more blood vessels into it. Even the tumor itself, it can start to invade to the surrounding matrices. We call extracellular matrices, and manage to invade into the blood vessels, perform we call distant migration, form secondary tumors. This is the process we call cancer metastasis. In fact, 90% of the cancer deaths are not because of primary tumor, but rather metastatic diseases. So, considering this model, how should we do using a similar concept? There are many strategies, like considering different types of cells. But one strategy that was proposed earlier, that they try to study the physical effect of the surrounding structure. Tumor surrounding structures are very, very heterogeneous. This is another example using glioblastomas. You can see the tumor cells colored in purple. They tend to migrate along certain physical structures. They are in the interface, such as vessel-stromal interface or periovascular space. These interface serves as a highway. The cancer cells, they take advantage of those highways to embark invasion and metastasis. So in order to recapitulate such interface, there are many studies, such as studies use collagen and collagen. They form an interface in between. Similar consideration using matrigel, the first layer matrigel, and adding the second gel of matrigel to form a cone shape, so that this tissue interface can be formed in the cone-shaped structure. There are also studies using polyacrylamide gel and by adding a layer of hyaluronic acid to form a sandwich structure for cells to migrate. But these cells are all studies on single cells. As you have seen, the tumor cell is actually a structure with multicellular cells. So this gives us a foundation that we wanted to do this tissue interface and studying how tumor cells interact with this interface. What you see is a model we developed. First, we use a collagen type 1 matrix to surround the in vitro tumor. And then we try to place this tumor in close vicinity of an interface. We allow them to invade into the surrounding matrix over 20 hours. So in order to do this, we use a microchip so that we can control the cells in the chip with very high content imaging capability. What you are seeing now is a time-lapse video showing how bladder cancer spheroids or tumors migrating across the matrix tissue interface. You can see this is a very invasive and looks like very lethal process. Certainly nobody wants to get such cancer. Using that technique, we are able to assess and analyze how this tumor progressed over time. So as you can see this figure, we can try to use confocal imaging to get upper level and the lower level. The upper level is marked with green dotted lines, and the lower level we mark it with purple dotted lines. You can clearly see very, very much two different distinct migratory behaviors at these two layers. Because we wanted to know how cancer cell interacted with the surrounding matrix. So we use this cyan color depicting the collagen. We can see the tumor cells actually doesn't grow much in the first 6 hours. Then over time, we can see a gray region marking that these matrices are actually depleted by the tumor cells. So that after this gray region shows up, these tumor cells start to migrate more and more. By quantifying the matrix we call void space, we can start to see at around 6 hours, we can see a significant void structure created. And then we can see the cancer cell migration start to catch up after that. We can also analyze the cells through this platform. So we have already seen different types of cancer cell migration, such as spindle cell migration or cobblestone cell migration. And we can also quantify them based on the trajectory and the path they have gone through. We also notice that the spindle cells is more directly moving as compared to the cobblestone type of cells. After 20 hours, we actually wanted to know what happened to our tumors after 20 hours. So we use fluorescent imaging. And by studying different stacks along a z-axis, we have seen a very clear void structure near the interface, but not so much as upper space. By rendering this tumor in 3D, we can see a very clear dome-shaped structure near the interface. This gives us a very, very encouraging result to know what happened. Is this really happened in vivo? Can we take advantage of this model for studying different drugs in the future? This is something we are working on right now. To give some concluding remarks, we are currently focusing on the study of the metastatic pattern of tumor, and we have demonstrated an in vitro avascular tumor model for assessing the therapeutic efficacy of cancer drugs. Right now, we are trying to leverage this in vitro tumor and to study how cells invade into the surrounding tissue interface. Currently, we have identified some very unique invasion features. So far, we have published 4 papers and over 10 conference proceedings. We were very fortunate in the early 2020, Professor Roger Ken, our international collaborator from Massachusetts Institute of Technology, has visited us so that we can learn with him in person. Finally, I would like to thank again to the Ministry of Science and Technology and the Young Scholar Fellowship, and also National Cheng University. Last but not least, I would also like to thank all my students for their hard work. Thank you very much.